Hello viewers, hope you all are doing absolutely fine. So welcome to my channel, that is Dhruv Rikhari Biology classes, and in this particular video we are going to start with unit number 3, that is plant physiology, and the chapter which I am going to discuss here is seeds, structure and its germination. The content of this particular topic is we are going to see about seeds, we will see about the classification and structure of seeds, we are going to see the germination and necessary conditions for seeds, and we are going to see some experiment on germination, that what are the optimum conditions for the growth of plant from seed. Next, type of germination, what are the types we are going to see, and germination in some common seeds, we will see the examples, and the seedlings. So let's start with the definition and by knowing basically what seed are. So in the previous video also we had discussed that ovule turns into the seed, and the ovule having embryo which sprouts into a new plant in suitable environment is known as seed. So basically seed is basically the physiological phase of a plant, and when it becomes a seed it undergoes a dormant period, which is an inactive stage of a plant, and the plant or the flowers, they get converted into the seeds and they are covered with the seed core, or they are present within the pods. Now seed is something which can remain in this particular form within the seed coat for long years, even for 100 years, right? So they remain under dormancy phase when conditions are unfavorable. So basically they undergo dormancy or inactive stage when the season of particular flowering plant or the fruit plant gets over. So they start forming a seed coat type of layer around, and they undergo inactive phase. So this particular phase is known as dormancy phase, and this basically happens when conditions are unfavorable. So right now I also told you that it can remain in this dormant phase for long years, even if the seasons are favorable and we are not placing this seed into the soil, they can remain dormant. So this is all about seed. Now seeds are different from grains, because grains are basically the fruit wall and the seed coat. Now fruit wall is basically the ovary, and ovule we know that's the seed. So basically the fused form of ovary and ovule is known as grain. Now I would like to know the difference between pulse and grain. So if you know the answer, write it in the comment section. Now moving ahead on the classification of seeds, the classification is basically on the type of seeds. So it can be monocotyledon or it can be dicotyledon. So monocotyledon and dicotyledons are basically the embryonic leaves, which are first formed leaves. So monocotyledons examples like grass, and dicotyledons are mainly like morning glory flower or morning glory plants. They are having a dicotyledon type of seeds. Next is about the seed vary in size. So the largest seed we know that it is a coconut seed. And next is on the basis of endosperm. So basically endosperm albuminous or X albuminous. So albuminous and X albuminous can even be divided into the monocot and dicotyledons categories. So now we can see the structure of seed and grain, and we can see here that the mango fruit, and within mango fruit there is a seed, and this is a bean seed, and this is basically the maize grain, which is a fused ovary and ovule. Now for bean seed, we can see here the structure of a bean seed having testa and tegmen. So testa and tegmen, so these two layers, if you can see, the outer layer is the testa and the inner layer is tegmen. So basically these two layers are forming a protective layer for the seed. Now what else we can see here is a small scar which is known as hilum or hilum, and over here we can see a micropyle end, and micropyle end is also helping in entry of pollen grains, and here its role is to imbibe. So basically when seeds are soaked in water, the seed coat gets ruptured. So rupturing of seed coat is basically when water is able to enter into the seeds. So this is the point from where the water is getting entered, and how that process is known as imbibition. Second is for the diffusion of gases. So basically this part is also responsible for respiration of seeds. Radicle and plumule are forming a root part and shoot part, and here we can see the plumules are having two rudimentary leaves, and plumules, as we know, that they are forming shoot part. Epicotyle is a part which is present between cotyledon and plumule. Hypocotyle, it is basically present below the cotyledon, and radicle is at the lowest part. So this is about seed. Now here we can see the example of maize grain, and this is an external structure which shows endosperm, position of radicle and plumule, scutellum which is a cotyledon of maize grain, and remains of style which is a female part. Now here is this dissected section of maize grain, and what we can see here, that fused pericarp and testa. The pericarp and testa are fused. So pericarp is the part of ovary and testa is the part of seed. So they are in a fused form, and within this part the endosperm is present, and epithelium layer is formed which is separating endosperm from embryo part, and within embryo we can see the coleoptile which is covering plumule, and radicle is covered under coleorhiza. This whole part is present within the embryo, and scutellum is also present here near to plumule and radicle. So this is about the seeds. Now moving on to next part, which is the difference between bean seed and maize grain. So what we had discussed from the diagram, this is the theory part. So two cotyledons appear, one cotyledon appear which is scutellum. Non-endospermic seeds. So basically bean seeds are non-endospermic, and the seeds of maize grain, they are endospermic. Plumule are large in case of bean seeds, and they have folded leaves, and they are having small plumule having rolled leaves. The structures are different for both maize grains and bean seeds. Hilum and macropiles are easily visible, as we had seen, and hilum and macropiles are not visible in case of maize grain. The seeds are covered in pods. So like in case of peas or in bean seeds also, we can see the seeds are covered within pods. And if you had also seen, or if you have seen the plant of Aprajita, in that plant also the seeds are covered in the pods, and the fruits and seeds balls are fused together forming single grain. So these are the difference between bean seeds and maize grain. This chart is very important for examination. I would like to request you all to please note down the things which are written here, and if you have any query, any doubt, write it in the comment section. And you can also follow me on my Instagram page, link is in the description box. So moving ahead, we are going to see the type of germination with examples. So now let's discuss the type of germination with example. So hypogeal and epigeal are the two types of germination. In case of hypogeal, the cotyledons are underground. In case of epigeal, the cotyledons are above the ground. Now in case of hypogeal, the epicotyle, which is the attachment of cotyledons and plumules, they elongate. The attachment of cotyledons and plumules, they elongate in case of hypogeal. In case of epigeal, the hypocotyle region of axis below cotyledon. So epicotyle is basically present near to the radicle or root part, and this part, in case of epigeal type of germination, they elongate. Now they are mainly monocotyledons. Hypogeal, they are mainly monocotyledons like grass, pea seeds, and maize grains. So these are the examples of hypogeal type of germination. In case of epigeal, they are mainly monocotyledons, and in case of monocotyledons, we had seen the first form leaves are, or the embryonic leaves, they are above the ground. Example is bean seed. So epigeal type of formation, you can see on my Instagram page. I had uploaded a story of morning glory plant, and I shared the first cotyledon after 5 days of sprouting the seeds. So you can get this point clear from that story, or if you want it, I will share you the picture. Next is about the seedlings. Now seedlings, they are basically the first stage of plant which is wholly independent of food stored in it. Now seedlings, when seeds are germinated and the cotyledons are formed, now from the embryonic leaves, they are able to uh, synthesize their food and they are able to store their food. So this is the state which is basically the seedling, which comes after the germination, where root can absorb water and minerals, whereas leaves start synthesizing food for the young plants and lead to the formation of mature plant that produce flowers and fruits. So this is all from this video. If you like this video, please hit the like button, subscribe to my channel, and also share it with your siblings and friends, and follow me on my Instagram page and on other social media links. Thank you for watching.